Hello, everyone, and welcome to today's webinar titled Standardized and Reproducible Solutions for Flow Cytometry Analytics. This webinar is part of the 5th Annual Drug Discovery and Development Virtual Conference, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Miltenyi Biotec. For more information about our sponsor, please visit them at www.miltenyibiotec.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Miltenyi Bozek, a product manager at Miltenyi Biotec. For a complete biography of our speakers, please visit the Biography tab at the top left of the screen. Miltenyi, welcome. You may now begin your presentation.
All right, hello everyone. Thank you so much for joining us today. My name is Miltenyi Bozek and I work as a product manager for Miltenyi Biotec. Today we'll be going through a presentation on standardized and reproducible solutions for flow cytometry analytics. So a little bit of fast facts, some information about Miltenyi Biotec. So we were founded in 1989 in Germany. Right now we have more than 3,500 employees worldwide. Across a number of different countries, currently have over 46,000, close to 50,000 different scientific references. We sell and provide greater than 17,000 different products, and notably, we're very proud to share this, that we have served and treated around 6,000 different patients worldwide using our technologies. So really have been involved heavily in the cell therapy, immunotherapy space. So our mission is really to advance scientific research in medicine by providing solutions for curative cell and gene therapy as well as biomedical research. So we are heavily involved in the fields of cancer, immunotherapy, neuroscience, stem cell, cardiovascular studies, and we translate that typically into clinical therapies in graft engineering, regenerative medicine, and immunotherapy. More specifically today, I'll be focusing on immunotherapy and flow cytometry analytics for that group of preclinical and clinical research. So we do provide benchtop to bedside products across a vast array of different types of portfolios. So anything from sample preparation using tissues to cell isolation, cell activation, cell culture products, plenty of cell analysis products, which I will be focusing a lot on today with our max quant flow analyzers and antibodies. But we also have a large imaging portfolio for high content imaging and light sheet microscopy. And all of these products can help to translate that basic benchtop research to the bedside for clinical applications in areas like graft engineering and immunotherapy, which I'll be focusing more on today. So at Miltenyi, we have 3 decades of experience in cell therapy. And we're very much dedicated to expanding our reach to new patients and innovating on those solutions that we provide to benefit researchers who are creating those next-gen therapies, especially for new clinical applications as well. So on this front, one of our key focuses is immunotherapy. We have decades of experience in immuno-oncology. The CliniMax platform is really one of the premier platforms in immunotherapy, including the CliniMax Prodigy for cell therapies. It's an automated integrative platform for T-cell engineering and also has been used in other application areas as well. The platform also has the ability to do customized applications if that is what the group determines they need. So a big focus on what I will be talking about today in relation to flow cytometry analytics is for CAR T-cell studies. So as of February of 2022, there are over 760 different studies around the world that are either active, enrolling, or recruiting just for CAR T-cell studies. So it's a massively growing field, and something that the whole world is working on together. So what is CAR T? So a lot of people know this, but CAR stands for chimeric antigen receptor T-cell therapy, and it's really achieved significant success in the treatment of hematological malignancies. So in recent years, fast-growing CAR T-cell clinical trials have really been growing and have really explored their potential for a number of different application scenarios. So as you can see on this graph, across a number of countries, the number of studies are growing and increasing year by year in the last decade or so of this being available. One of the questions, though, that we get pretty often at Miltenyi is, you have this, great automated platform to produce autologous cell products. So why is the cost of manufacturing still so high, given that you have that automation? It's a very good question, and it's true. It's still extremely expensive to produce one lot of an autologous T-cell therapy. So where is that coming from? Looking at this particular study, labor far and away compared to just like capital equipment, materials, consumables, other types of costs. Labor is far and away the highest cost in generating a cell therapy for the patient. There's of course a whole suite of analytical processes that come into that labor component for autologous cell therapy production. And this includes that flow cytometry analytics. So today I'm going to get a little bit more into ways that we can streamline and make this a lot more cost-effective, faster, easier for that analytics qualification process. So it's also pretty important when we're thinking about that qualification characterization process for cell therapies using flow. It's important to understand the critical path to the clinical space in both the upstream of submitting an IND, but also past that. So as you're working upstream of submitting an IND, there are a number of major hurdles to overcome during each of these different steps of development, tech transfer, and then getting to the GMP space. So, if there are very manual or difficult steps, it really can be a bottleneck to get all the way to an IND. And without appropriate analytical methods, it can really be difficult to have, those early stage feasibility experiments or studies. But once you achieve those, you can scale up and do, part of the tech transfer at that point. But that particular process of transfer and engineering can be a bottleneck, and often flow cytometry is a critical part of that tech transfer. The transfer to GMP as well requires specific methods to be developed for a smooth transition. And if you're lacking those methods at that state, this can really delay your process qualification, your in-use and shipping stability studies as well. And you need to get to those long-term stability studies in order to, submit your IND properly. So today I'll really be focusing on some of those methods to mitigate delays and accelerate the time to IND and to market by showcasing some solutions that Miltenyi has specifically in the flow cytometry analytics space. So it's important to understand the relevance of reproducibility. So there's a study done back in 2015 that looked at, all the different components of reproducible and irreproducible research, and I'll showcase that a little bit later when it comes to reagents. But it's important to keep in mind that irreproducibility of results can cause tremendous, economic damage when it comes to companies and, just time lost, which really leads to, longer drug development timelines and, just more of a headache for everyone involved in this particular area of research. So, the types of categories for preclinical irreproducibility, you have, and this comes from that same study, you have things like biological reagents and reference material, the study design, the data analysis reporting, and then laboratory protocols. And what they found is that actually, over 70% of that irreproducibility can totally be influenced by the experimental setup and not necessarily the study design itself. So ton of opportunity in that space to deliver on better types of biological reagents and reference material and equipment in order to mitigate those types of issues. So historically, new cell products have originated from academic environments, and they're developed in basic research using very well-trained scientists. Translating this, though, into a commercial manufacturing environment can be very difficult. You need to do scale up of, very manual, complex procedures that require, basically humans to be perfect. And that's obviously a, very difficult goal to be met. So, what we strive for is to remove the need for the human operator excellence and translate that to more standardized, robust, integrative processes with automated workflows that can just increase productivity much more so. So really the paradigm is to, integrate, automate, and de-risk this process. So, when you're moving finally from the GMP process to commercial production, you have to keep in mind at each one of these stages, there's really a critical phase at which, you can't go back and you can't get rid of those variations if you're seeing them, later on within this process. So if you can reduce variations early on, you're really increasing the chance to successfully transfer your process to the clinical phase. So a little bit of what I'm going to be going through today is what we call IPC/QC in cell manufacturing. And this is a place where flow cytometry standardization is really key. So a little bit about the process of CAR T-cell infusion. You have a number of different stages, and I'm not really going to go into the details here, but it's important to keep in mind that when you're doing an autologous cell therapy, you have this apheresis to manufacturing back to infusion stage. And at each one of these stages, you can be using a number of different methods, including flow, to assess product composition, product sterility, the process status, how well did your transduction go? You're looking at compliancy and timing. And so all these aspects in IPC/QC are really crucial for this whole process. Those quality control steps have to be validated and qualified, and they're also necessary in order to have the final product for release back into the patient. And so, at each one of these steps, you need to have, flow analytics to assess, the quality of your product during in process, and then at the final stage of QC in order to release that cell therapy product back to the patient. So today, specifically for CAR T-cell studies and understanding how we can, mitigate sources of variation, we'll be looking at 5 different sources of variation that contribute to it in flow cytometry, specifically for cell therapy in the immunotherapy space. So donor variability is the first one. It's obviously very patient-driven, pre-treatment driven, and collection driven. And, there's only so much you can really control, when it comes to donors. Also going to go into a lot of depth on reagents, lot-to-lot differences, specificity, sensitivity of, antibodies that are used in flow. We'll go into some solutions that Miltenyi has innovated on when it comes to instrumentation of our max quants, innovation when it comes to analysis and reporting, things called express modes that I'll get into. And then, very briefly, just on the support we can provide when it comes to protocols, when you're working with, non-optimized procedures and trying to get rid of, manual handling. So the first source of variation I'll touch on, and, really the briefest, is just on donor variability. It has a huge effect on whether or not a cell product will have a reproducible and consistent performance. So in that table on that you're looking here, or that we're showing, is you're looking at different populations of subsets of blood cells that did meet the dose criteria for a patient or did not meet the dose criteria for that patient. So, there's only so much we can do, and it can potentially be mitigated by cell processing steps like cell enrichment. But beyond that, there's not anything particularly direct that we can do with flow cytometry to improve this. Going into that next source of variation in flow cytometry analytics, we're going to look at reagents now for a bit. So it's estimated that the U.S. preclinical research spend in categories of errors that contribute to irreproducibility is extremely high, right around 50%. If we look deeper into those sources of irreproducibility, you can see that a majority, almost 40% is spent, the majority parameter, 40% is spent on reagents and reference material. So, no doubt you could improve, the training, the design of the study, and increase quality control to add to that factor. But the very fact of not having good quality, reagents and reference material could really improve, society-wide the quality of our research and the spend and the money we, invest into it. So, it's a huge issue, and it's something it's estimated something that over $14 billion a year are wasted and could potentially be improved on working better in improving that quality of reagent and reference material. So a lot of this in flow, not surprisingly, is due to the antibodies in particular. So they're very much a large cause of irreproducibility that we see in preclinical research. So back in 2008 through 2012, a number of studies were conducted specifically to look into these causes. And by 2014 to the present day, there are a number of active groups, of which Miltenyi is a part, to really work on improving these levels of nonspecific and irreproducible results due to antibodies. We've certainly made progress, but as a whole, there's always a ways to go and a number of ways to improve. So what are some of the factors that can result in irreproducible data in flow cytometry? So there are many, and some of the major categories are listed here. And some of these probably don't come as a surprise, but taken all together, it really lends itself to a really complex type of analytics that needs to be controlled better. So with antibodies, a lot that are out there on the market are very poorly characterized. They may not have sufficient sensitivity. Lot-to-lot groups of antibodies can vary widely in performance and how they bind. There can be a lot of background noise. They may be non-specific to the target that you're actually trying to understand. The manufacturer may not have a lot of information on it. So you as a researcher may need to do a lot more investigation on your end to get more data out of it. in relation to that, you may not know what the performance of the antibody is like. So things like how does it perform under fixation, could be really useful to know before, buying a particular antibody and using it in your experiment. The protocols that are provided with the antibodies may not often be accurate for the particular antibody that you're going after in your panel. And then finally, there are so many other variables that can be included in the manufacture of antibodies that just require, different types of controls along the way of manufacture. So today, what we're going to discuss when it comes to reagents is, how does Ms. Miltenyi help to, overcome these reproducibility issues from the reproducibility crisis that specifically relate to antibodies. So at Miltenyi, we have worked through 3 different solutions when it comes to reagents. We use advanced technology through our reaffinity recombinant antibody technology. We have proper validation of those particular antibody reagents, and also include thorough documentation for all of our different recombinant antibodies. We have the ability with recombinant antibody technology to innovate, by ourselves in this flow space, but we also work with, researchers around the world to do that proper validation of those antibodies, and then, work with others for thorough documentation as well. So it really, in a way is kind of a team effort. So at the moment, before we get into, recombinant antibodies, it's important to understand that there are, currently 3 major groups of antibodies that are used in flow. So this chart kind of highlights just general high-level overview of those different ways of manufacturing antibodies. So polyclonal antibodies are a mixture of heterogeneous antibodies. They often recognize several different epitopes, and you may see a stronger signal. They're made from purifying the serum of immunized animals, but the issue with it is that there's very low lot-to-lot consistency, and so for that reason, it's really not the right antibody for flow at all. So the next generation of antibody, monoclonal or hybridoma-derived antibodies, are a much more homogeneous group of antibodies. They recognize one epitope and so have much higher specificity than polyclonals. They're purified from clones of immortal hybridoma cell lines, and so they do have a higher lot-to-lot consistency, but really still, aren't the best solution out there when it comes to flow. So recombinant antibodies are genetically defined antibodies. They're defined very much on the DNA level, and they recognize one epitope with the highest amount of specificity. They're genetically engineered and produced in a very controlled and defined expression system, unlike monoclonal antibodies from those hybridoma cell lines. And so they really offer that highest lot-to-lot consistency for flow cytometry analytics. So, most researchers nowadays, use monoclonal hybridomas or a combination of recombinant antibodies. And this is really, the way that, flow has gone, much more recently. So let's understand a little bit more about the differences of hybridoma-derived antibodies versus recombinant derived antibodies. So hybridoma clones are generated from antibody-producing B cells, and they need an immortal myeloma cell line in order to continue to pump out antibodies. So those cell lines are generated for a long time and can generate spontaneous mutations and changes to the antibody due to genetic drift. All this leads to lot-to-lot variability and decreased purity in the final product. So recombinant antibodies, on the other hand, are quite different in that they have a very defined genetic sequence. They're programmed to express that particular antibody product. And because they're not cultured in B cells, they have a much more controlled culture system. They don't have the same types of contaminants that you see in hybridoma models. So Miltenyi offers reaffinity technology. It's an innovative and growing portfolio space where We offer the largest portfolio of recombinant antibodies for flow on the market. So what is this specifically? So since 2012, we've been offering this and innovating on it. So what you're seeing on the left here is a model of the antibody where we took advantage of genetic engineering. So the antibody variable region shown in orange is typically derived from a hybridoma clone that was validated on flow. And so we take that sequence and engineer it into the recombinant form of the antibody. So then you can see the backbone in purple. The constant region is an IgG1 backbone, so it's universal basically for every reaffinity antibody. And in part of the backbone, this particular region is the Fc region, which we have actually engineered a mutation in it so that there wouldn't be excess Fc gamma receptor binding. So you'd really have diminished background signal from non-specific binding more easily. Getting into the quality of reaffinity antibodies. So here's an example showing mass spec data where the recombinant antibody shows a much more pure product in mass spec compared to the hybridoma clone. So this pink star shows the contaminating light chain that you get in monoclonal antibodies, whereas reaffinity antibodies typically do not have this. So you have a much more pure product with reaffinity antibodies. Here I'm also showing more information on that Fc receptor mutation that we were talking about on the backbone. So you're looking at immunoassay results where we compared the binding of the reaffinity antibody in light purple. So these light purple small bars here versus the mouse monoclonal antibody in dark purple, which are recognizing CD144 of 5 cellular human Fc gamma receptors. So you can see significantly lower binding of that recombinant antibody than the hybridoma to those Fc gamma receptors. So why would we even really care about this aspect of antibody reagents? So this is just one example of some customer data, but it illustrates it pretty well. So the issue is really background staining. The area in red that you're seeing right here shows non-specific binding from that hybridoma clone. The other hybridoma clone shown, for example, doesn't have, that necessarily that same level of background staining without the Fc block. And you can see for reaffinity, you don't even need the Fc block and you get much less background staining. And so even with an Fc block, with that hybridoma clone, you're still getting, a fair amount of background staining, whereas the reaffinity clone performs, essentially the exact same whether or not you have that Fc block. So, really makes a difference when it comes to the quality of the data that you're getting out from a recombinant antibody during flow. One of the other major advantages to recombinant antibodies is that universal isotype control. So you saw that the recombinant antibody backbone contains that same IgG1 across all recombinant antibodies. So the benefit to this is that, many different hybridoma antibodies within, just a single panel could easily have tons of different types of backbones, thus leading to potentially, much more complex experimental planning and inventory management, where you really only need one type of, isotype control for your panel if you're just using recombinant antibodies. So really making a much simpler experiment and inventory management a lot easier for yourself. So I also wanted to show the level of quality that we go through for our antibody production process. So here's an outline of that purification process. First, we have really strict quality control of our starting materials for both the antibody and the fluorochrome that we've, chosen to do conjugation for. So they're conjugated. And then, at that stage, we do a very thorough purification process to get rid of unbound fluorochromes and any excess, antibody components. So this really helps to ensure that we have a highly pure, highly consistent product. And we do final functionality testing, which I'll get into a number of examples here, on those primary samples to really ensure that the antibody is performing accurately and precisely as intended. These functionality tests are often done on a mix of dissociated and processed samples to really attempt to cover the different types of samples that many researchers use. And of course, these studies are always performed with a titration. So you're really getting a much more, lot-to-lot consistent product than you would, from potentially other manufacturers who don't go through this type of process. So here we have an example, and this is just, now I'm getting into a bunch of different examples of the types of tests we do on our antibody products. So here we have an example of a bunch of different lots of antibodies that perform identically. So we're making sure that, lot to lot, the staining performance is nearly, identical between all these different types of lots. This also just helps to ensure that our customers don't have to perform titrations every single time they use the, exact same antibody. And we also have a very easily accessible C of A portion of our website where, after purchase, you can look up your respective lot and get your C of A and more information on that particular product. So I'm going to go a little bit into antibody specificity testing. So antibody specificity is really a critical point because you need, a highly specific antibody to really understand, the level of staining and the type of cell product that you have for your, CAR-T or CAR-NK product. So we need to be able to ensure that the target that we're actually going after is actually what the recombinant antibody is targeting. So what are we doing to ensure that that specificity is real? So we perform a number of different suitable experiments like counter staining to ensure that the antibody is actually binding to the antigen of interest. We perform a knockout of the target protein and epitope competition assays. And for specificity, we perform siRNA knockdowns, stimulation of cells, overexpression of the target protein, as well as binding of that antibody to purified antigen using like a latex bead coating. So we do a lot of experiments around specificity to ensure that quality. So I'm going to quickly go into how we get a little bit deeper into the knockout protein assays. So we generate cell lines where we knock out the protein of interest and compare performance of the antibody of the knockout to the wild type, and then look at its specificity. So you can see in the wild type cell line, the target antigen is present, it's shown in red, and in the knockout cell, it's gone. And we're also doing this by flow as well. So this is just nice because, the benefit is, you have less validation effort and can be a little bit more confident on that antibody quality. The second test that I'm mentioning that we perform is the epitope competition test. The clones we make at Miltenyi that are recombinant start with, they're qualified by the word RIA and then a number. So they may not actually, you'll see that and you may not actually recognize, the clone that you're used to in terms of nomenclature. So even if the epitopes that they target are meant to target the exact same antigen as the, monoclonal hybridoma antibody that you're used to. So what we do is we perform the epitope competition assay against established hybridoma clones to ensure specificity. So the way we do this, it works like a blocking experiment. So in regular staining, that the antibody is conjugated to a fluorophore and it will show 100% signal. In a competition or a blocking assay, we use a pure version of the antibody with no fluorochrome to block that epitope of interest. Then we dose in the hybridoma, comparable clone conjugated to a fluorophore, and that's, the comparison that we're actually trying to compare the recombinant antibody against. So if it shows 100% signal, it means that there were no overlapping epitopes. Now, if there's a spectrum of positive to completely negative signal, it would mean that they have, partially overlapping or completely overlapping epitopes. And this would be a way that you could, look up, your hybridoma clone of interest against, the RIAs that we provide. So you can find this information on our website for each of our antibodies, and you'll see it on the product page, information on, completely overlapping epitopes to completely different epitopes with clones that you may be, already familiar with. So getting into one of the final aspects of antibody quality that I'd like to talk about is sensitivity. We want to be sure that the antibody that we're looking at is actually sensitive enough to meet a variety of different applications or many different needs that you may have across different types of research. So here are some of the different types of assays we use to establish sensitivity. Performance is tested in flow, also tested in microscopy, and sometimes in mass cytometry as well. It's also compared to alternative hybridoma clones that are out there on the market, and we'll display this data on the product page of the website. We also test it against fixation and then show data in multi-parameter flow cytometry for just more complete information. So that performance comparison in flow, it's just very basic tests that we do for recombinant antibodies, where we just do head-to-head comparisons of, other provider hybridoma clones against the RIA clone and display it so that users can find it, very easily on the website and see if it's something they're interested in. All right, so now I'm going to move on to instrumentation and some of the innovation that Miltenyi has done in the flow analytics space to help, solve things around operational tolerances, manufacturing tolerances, and just get into a little bit more detail on that. So there are a number of tools and strategies we use to eliminate human error, data variation, and try and deliver reproducibility in simpler use for operators of instruments. And this is really, a key goal of ours at Miltenyi. So one of the aspects that we build into our max quant flow analyzers is called smart gain technology. So this aspect of the max quant really allows users to have multi-instrument harmonization. So in practice, max quant instruments with smart gains allow you to run the exact same instrument parameters from like a master max quant, in theory, at multiple sites around the world because of the standardized instrument settings. So the challenge really here for a lot of users is getting reproducible data, from one site to another, or with even one site to another instrument at the same site. So to really deliver on reproducibility, you need both an intra-instrument performance characterization and also inter-instrument performance characterization. So how can we enable this? So to ensure reproducible performance over time, we need to consider and understand that there are intrinsic differences of each detector in flow cytometers on each channel and each detector on each device, specifically when looking at PMT gains on flow cytometers. This is very much inherent just with the design of a lot of flow cytometry instruments and not something that you can, necessarily get away from. However, these detection channels behave linearly, and so, you can calibrate them against one another and get more consistent reproducible data. So knowing this and working with calibration beads, you'd use a master max quant device with a reference and control material with a known MFI. So to harmonize those instruments with different intrinsic properties across sites or even within the same site, you can use the same instrument settings from one device to another. So this example histogram shown here shows the reproducibility of smart gain technology with the same sample donor across 5 different devices. And the overlay really shows the ability for us to get consistent data and for instruments to perform similarly. So you can think of smart gains kind of like as a lighthouse, and the beads, act like that lighthouse, that stable reference point that can be calibrated against. So in calibration beads, we have known fluorescent values that should reach, a specific median intensity. So, device A here needs a PMT voltage of 400 to reach an MFI of 600. So after daily calibration, the software and the instrument gains are automatically adjusted. This is possible because the instruments are calibrated to known and established MFI values. Device B may need a PMT voltage of 430 in order to reach the same MFI value of 600. So what we've done is we've harmonized this with smart gains with those instrument settings that are established after calibration. You get consistent results with the simplicity of a universal instrument setting. So all you're really doing, very simple, is you have to save the data from one instrument, transfer it to another, and then continue on in your assay. So here's just some sample data that was generated using a master MaxQuant with 4 other MaxQuant instruments with a standard 8-color immunophenotyping kit along with an express mode, which I'll get into in a bit. You can see that the data generated across many of these different many different subsets of cell types is giving very similar and really consistent and reproducible. Another aspect of a way to mitigate variation in data and flow is our IQ/OQ services. So we do offer these types of services for instrument qualification and really allow you to maintain that GMP standard so that you can get official documentation and really be covered there. So now I'm going to get into another source of variation in flow, which is analysis and reporting. And so this is, very much a human factor in flow, where you hear people say, flow is sometimes an art, but we do have a way to really make it standardized across users with this feature called express modes. So express modes, allow you to kind of get rid of some of this sample-to-sample variation. So typically, you would need to, change the gating when done manually, even for 2 different types of donors. And so you can see in this particular example, you're not getting, even though it's the same exact gate, it's looking a little bit differently when you're actually doing your gating strategy. So express modes really allows you to adjust it based on algorithms that can really help to avoid variation caused by that human factor. So here you're seeing that algorithm adjustment because of 2 different donors in the express mode. So express modes, with a little bit more detail now, they're an automated approach to flow cytometry analysis. Express modes provide standardized algorithm-based data analysis to really reduce human error, allow you to have reproducible results, and provide a standardized analysis truly based in mathematics, and allowing you also to simplify, SOP-based processes. You can really think of express modes as software modules with predefined settings that allow for a much faster and less error-prone analysis. So users, when they're operating a MaxQuant typically manually, have a work time of somewhere around 17 minutes a panel, whereas using express modes, you can get close to 30 seconds per panel and really streamline that analysis process. So when you're actually looking at the reproducibility of manual data analysis compared to express modes, the variation that you see in manual handling of gating, of just individual users is really much higher and can be mitigated by using express modes. So you're really, eliminating that human factor when it comes to flow gating and really, that major headache that a lot of people tend to have in this area. So there's also 21 CFR Part 11 enabling compliance, and you can think of this also as a module that can be enabled to give you compliance on MaxQuant instruments. So it really benefits the types of users who need that high quality electronic record and signatures for documentation. It also makes it much simpler for submission to relevant regulatory authorities. The 21 CFR Part 11 enabling module does allow for an audit trail. So this means that you have time-stamped audit trails for all data files, which is recorded at the creation, modification, and storage of those files. In addition, it's also a closed system with electronic signatures. So you have a secure user login with password-controlled inactivity timeouts, renewals, and attempted sign-in failures. We also have advanced user and role management systems. It's a way, you can have this optionally to connect your customers to your own IT system. And finally, there's an electronic record with comprehensive reports that include all the necessary information in a PDF that really is forgery proof and eliminates, the concerns there. So Max Quantify Express modes really can be as simple as press and play. So the way it looks like on our instruments is you have, you're basically just enabling the type of, acquisition that you're doing. And so here analysis, and then the mode. So you're choosing, the particular express mode, which I'll get into the different types we have in a bit. So super easy and standardized right here. And then all you're doing is pressing play and walking away. So the types of examples of express modes that we have are, kind of across the board. This is just one, and probably one of our, most highly used one, called the immune cell composition. It allows you to look at the array of different immune cell types within a population. It's a 9-color panel that has, automated gating and, can be used with standardized reagents that I'll mention in a bit. Really allows for that plug and play approach to flow analytics for cell therapies. So you'll get readouts like, this large table of statistics, which includes cell types, defined population percentages, frequency over total of that cell type, absolute cell count, which comes automatically because we have volumetric cell counting, and frequency percentage among viable CD45 positive cells. So this one, as I mentioned, this express mode, the immune cell composition, is one of our most commonly used modes for incoming evaluation of blood products, and very heavily used in the cell therapy space. Another express mode that's commonly used in IPC-QC is the transduction efficiency express mode. So again, you'll see dot plots with the gating strategy clearly defined in that output and report for transduction efficiency. And you'll see the same statistics table with populations of interest. And then this time the product release information is also clearly defined at the bottom there as the viable amount of CAR positive, CD3 positive cells. And all of this can be exported to PDFs and Excel sheets as well. So this is just a brief look at just our CAR T express mode package. You can look at things like deep phenotyping, persistence, exhaustion, activation, proliferative ability, as well as some controls, and then analysis of malignant B cells for people who are in that space as well. We do have the ability. We have other express modes that come automatically with MaxQuants, and then we also have the ability to do custom express modes for any flow panel that you might be interested in as well. So just a quick plug, we do have a new product, Stain Express, which are pre-formulated dry antibody cocktails based in reaffinity technology that are really designed for IPC-QC and work perfectly hand in hand with express modes. So, a current workflow for, doing these types of panels in IPC/QC would, be a lot of manual hands-on labor. But with Stain Express, you really have that ability to just add your sample to a barcoded tube, maybe add your lysis buffer if needed, leave it for staining, scan it on the instrument using the express mode, and then just, plug and play and let it go. So going back to that IPC/QC process in CAR T-cell manufacturing, the two different Stain Express panels we currently have, immune cell composition and CAR T-cell transduction, labeled as 1 and 2, can be used at various stages along your production process for your cell product. We also have CAR detection reagents. So depending on your application, you can choose one of our CAR detection reagents and dose it into that transduction cocktail to really kind of understand a little bit better that efficiency of your product in the manufacturing process. So we have antigen-based CAR detection reagents, as well as idiotype CAR detection reagents against the FMC63 region of the CAR. And so we have a variety of products conjugated directly to biotin or to fluorophores directly for the idiotype CAR detection reagent. So overall, this IPC/QC CAR T cell process, we have, 3 major groups of products that really allow you to get that standardized, consistent analysis. So, from the software modules, express modes, to Stain Express, to our CAR detection reagents, you really have an optimal IPC/QC setup with a robust measurement, that unified antibody backbone, and a flexible CAR application depending on, the type of marker that, you might need for your particular cell product. So very last, source of variation in flow cytometry analytics is protocols. So we took this from the Clinical Lab Standards Institute protocols that were released. We are able to, help you along all the different steps of assay development that could cause variation. So, understanding what your intended use is, determining, the type of instrument configuration you need, panel design, and then your analysis strategy. So, Miltenyi, we have the ability to help you for everything along this way, from sample processing to sample analysis, your process requirements, where, we can help you with customized express modes, the platform component compatibility, so we can work with you, on the instrument platform that you're using. And then, of course, we have service and support that can work with you every step of the way, and also help to iterate on application development as well. So in summary, the main sources of variation in flow cytometry analytics, Miltenyi can really help you mitigate them by a number of different factors, from, our reagents with the reaffinity antibodies, instrumentation with SmartGain technology and IQ/OQ, our software packages and modules with express modes, which are algorithm-based data analysis as opposed to manual. And then finally, we have ways of, improving your protocols with automation, customization, and then application support. So all these different concepts, all these different standardization concepts will really assist you in mitigating those sources of variation during those critical phases of process development where you can't go back and change. So Miltenyi, we really, work with our customers as partners to get you to, not have to have issues in those critical phases of, development to IND, all the way through commercial production. So thank you very much. I am, very grateful to Labyrinths for letting us present. And please, if you have any questions, feel free to email me at emilybo@miltenyi.com. And, I can answer the question or can, facilitate a conversation with someone who who may be more of an expert. So thank you very much, and hopefully we'll talk soon.
I want to thank you again, Emily, for your time today and for your important research. And before we go, I want to thank the audience for joining us today. And I'd also like to thank our sponsor, Miltenyi Biotec, for sponsoring today's webinar. Questions submitted today and during our on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until February 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.